Okay, welcome to a run through of the OCR A-Level Biology Biological Processes paper. This is a sample paper that was produced for the cohort who are being examined with the new exam in June 2017, which has a new style and includes content from AS and A2 and is examined very synoptically. So we start with some multiple choice questions. So it starts off with a relatively simple multiple choice question which asks which statement explains the significance of mitosis in the development of whole organisms. A few absurd suggestions. So it says in D, budding in yeast is an example of mitosis, which it is, producing new multicellular organisms, which it definitely isn't. It also comes up with sex cells are produced by mitosis, which they're not because they're produced by meiosis, which leaves us just with A and C. And the answer here is A, that mitosis can be controlled at certain points in development, which will change body plans. So the answer here is A. Then leads on to which graph represents the countercurrent exchange system in fish gills. Well, two things to exclude instantly are A and C. Remember, countercurrent flow involves blood flowing in an opposite direction to the flow of water. And this is over the gill plate on the gill lamellae. And the reason why this is so efficient is that it maintains a concentration gradient over the entire length of the gill plate, so that the blood is always encountering water with more oxygen in it. So oxygen is always diffusing into the blood from the water. And here is distance along the gill plate in B, and the blood is going to be flowing in this direction along the gill plate, and the water is going to be flowing in this direction. So as the blood travels along the gill plate, the percentage saturation of oxygen will rise until it's at almost the same level as the oxygen as it's coming in. So the answer here is B. Go B. And on to the next paper. Okay, so now it says cells require vitamins and minerals in order to function correctly. These need to cross the plasma membrane. Then it carries on. Vitamins are either fat or water soluble. Then they give you A, D, E, and K, which are fat soluble. So this question is really asking about the concepts of how things cross membranes and why some things are unable to diffuse across the membrane. And the reason why some substances can't diffuse across the membrane is because they're not soluble in fat. Now these substances, A, D, E, and K, are fat soluble, so they will dissolve in the phospholipid bilayer, so they can diffuse straight across. It then asks which of the following combinations enter the cell by facilitated diffusion. Now, facilitated diffusion is assisted diffusion. To facilitate is to assist, so it's by using some form of a protein channel or protein carrier. It's still diffusion, which is the movement of substances down their concentration gradient. So in this, it's first of all thinking about which things can you exclude. So vitamin A is fat soluble, so it's definitely not going to be point A or point D. And it refers to calcium atoms, whereas in fact, remember, it's calcium ions that are going to travel across. So the answer here is C. Now, animals often receive different stimuli from their environment. Now, if those different stimuli if they're receiving different stimuli from their environment, then those stimuli are coming from different receptors. The synapses can manage multiple stimuli, often resulting in one response. So this action of the synapse is an example of, well, it's a spatial summation because it's integrating stimuli, messages from many different receptors at the same time. Onto a calculation question. Well, you don't actually have to do the calculation, it's fairly obvious what the answer is. So the kidneys of a healthy individual filter 178 decimeter cubed of fluid from their glomeruli into the renal capsules. However, only 1.5 decimeter cubes of urine is produced. Now, what percentage of the filtrate is reabsorbed? So the way you'd have to do this would be to take 178 minus 1.5, divide by 178, and multiply by 100. And this is D, which is... The mistakes people make here is they miss the point of reabsorbed. They miss what's going on there. The following mechanisms are used to move water through plants, so diffusion, osmosis, mass flow. Then it asks you which flow correctly identifies the mechanisms used at each point of the transpiration stream. The transpiration stream is the movement of water from the soil out of the leaves, and it's caused by the evaporation of water and cohesion, tension, and the root pressure created in the root cells. So my recommendation with this is always to look at what's in the first column and what's in the last column. Start with that and then think about excluding certain answers. So how does it move into the root cells? Well, it definitely moves by osmosis, and it definitely doesn't move by diffusion, so we can rule out B and C. Then if we look at the final column, how does it move out of the leaf? Well, hopefully you should remember that it moves out of the leaf by diffusion because it's moving down its water potential gradient into the air spaces in the spongy mesophyll and then out through the stomatal pores into the atmosphere. So this leaves us with the answer A. Okay, next on to a question about enzyme inhibition. So citrate synthase catalyzes the conversion of oxaloacetate to citric acid in the Krebs cycle. So this is a question which is combining your knowledge of respiration, which is a second year topic, with enzyme controlled reactions, which is a first year topic. You should remember that oxaloacetate, so that will be the substrate, and it's being converted into citric acid, which is the product, and it exhibits end product inhibition. So which of the following is the correct description of citrate synthase? Well, citrate synthase, the inhibitor is going to be the product, which is the citric acid, so this gives us A and B. Now, it tells you this works in the Krebs cycle, so we should be aware that the Krebs cycle occurs in the matrix of the mitochondrion. So this just leaves us with B. So which of the following describes the process that happens during repolarization of the neuron during the action potential? So remember, with a neuron, you have that it is polarized by the movement of sodiums and potassiums. So the sodiums are moved out, the potassiums are moved in. This is powered by ATP. When you have an action potential, you reach the threshold value, the voltage gated sodium channels open, the sodium floods in, and this depolarizes the membrane. Next step is the repolarization of the membrane. Is the potassium channels open, and the potassium that you've actively pumped in then flows out again down its concentration gradient, and that efflux of potassiums repolarizes the membrane. So to describe that process, well, the membrane potential is decreasing because it's being repolarized back to the resting potential of around 65 millivolts, and in order to repolarize it, you have opened the potassium channels so that the potassium is then flooding out again. So the answer here is A. So then onto an unknown solution of a single sugar was tested. The results were recorded. Colours observed. So the Benedict's test is blue, which means it is not positive, whereas for a non-reducing sugars test, it goes brick red. Now remember, the non-reducing sugars test involves boiling it with hydrochloric acid. So identified the unknown sugar. The answer here is D. It is sucrose, because that is the non-reducing sugar, in that fructose, lactose and glucose are all reducing sugars, and they would all test positive with Benedict's. Okay, so now on to an anticodon sequence of 5 successive tRNA molecules was analysed and found to have the following percentage base composition. Which row shows the percentage base composition of the template strand of the original DNA molecule? Well, remember that when you have a DNA strand, the template strand will be transcribed. So if this was a template strand here, then that template strand will be transcribed into messenger RNA, so that the DNA triplets would be transcribed into messenger RNA codons. Then the tRNA has anticodons which are complementary to the messenger RNA codons. So you've got tRNA which is complementary to the mRNA, and the mRNA has been made by the transcription from the complementary DNA nucleotides. So that the anticodon sequence will be exactly the same as in the DNA, except the uracil will be replaced by thymine, because you've got the tRNA is complementary to the mRNA and the mRNA was complementary to the DNA. Therefore the DNA strand will be exactly the same as the tRNA, except remember there is no uracil in DNA because the uracil is replaced by thymine. So the answer here is A. So this question is about the control of temperature in an endotherm. So it gives you heat flow through the skin of an athlete during vigorous exercise starting here, and then you can see that over R, the heat flow, the heat's dissipated from the skin in watts per meter squared, this falls over the first 3 or 4 minutes. Then there's a rapid rise at S, and then a little decline over T. So they're asking you to explain what's going on. So which row gives the best explanation of the stages? Well, point B, the blood is directed away from the skin and towards the muscles. It's then directed towards the skin, which is why the temperature goes up, where there's more heat radiated from the skin. And then at T, it flattens off again. So you're going to get the decline over R, the rise at S, and the steadiness at T. So the answer there is B. So this question looks at which of the following are interventions. And the crucial point here is interventions in the control of blood glucose concentration. So statement 1, insulin injection, well, that's definitely going to control blood glucose because it's going to be, and it's definitely an intervention. Regular cardiovascular exercise will reduce blood sugar level, but is not an intervention. And glucagon injection will increase blood sugar, but is not a regular intervention which is carried out. So unfortunately, the only answer here is D. Which of the following statements are true? Microtubules are part of the 9+2 formation in bacterial flagella, which they're not. Microtubules can be prevented from functioning by a respiratory inhibitor, which they can. Microtubules are involved in moving chromosomes from the equators to the poles during mitosis, which they are. So the answer again is C. So blood vessels are adapted for their function. Which of the following statements are true? The walls of the arteries near the heart contain elastic fibres so that they can stretch and recoil to maintain blood pressure, which is true because they have to have an elasticity as the blood spurts out of the ventricles and so that they can expand. The walls of the venules contain very little muscle, which again is true, and the walls of the arteries contain a lot of muscle fibres, but then it goes on to contract and generate pressure. So they do contain a lot of muscle fibres, but it is not to contract and generate the pressure. The pressure is generated by the ventricles, not by the muscle in the arteries. So the answer here is B. Okay, next, phospholipid bilayers play a crucial role within plant cells. Which of the following statements linked to the importance of membranes in plant cells is or are true? So the ATP synthase embedded in the thylakoid membranes maintains the chemiosmotic gradients. Well, it doesn't maintain the chemiosmotic gradient. The chemiosmotic gradient is maintained by the flow of electrons along the electron transport chain. The ATP synthase dissipates the chemiosmotic gradient when the hydrogen ions flow through it to generate ATP. So statement 1 is not true. Statement 2, phospholipid bilayers within the chloroplasts are impermeable to protons. Well, they are, otherwise you would not have a chemiosmotic gradient. And the thylakoid membranes contain electron transport proteins. That's definitely true because the flow of electrons between the photosystems is used to move the protons from the stroma into the thylakoid interior. So the answer here is definitely C. So on to an ECG question. So this shows the ECG pattern for a single normal heartbeat. So the P wave is atrial systole, the QRS is ventricular systole, and the T wave is ventricular recovery. They then give you two ECG traces, one for a person with a normal heart rhythm, which is this one here, and the one for a person with tachycardia, which is this one here. Calculate the percentage increase in heart rate for a person with tachycardia compared to the person with normal heart rhythm. So for this, you've got to look at the time taken between a heartbeat. So with this, you just measure the the distance between where the points recur, and you'd convert that into a time, and then you'd divide 60 by that distance in seconds to give you beats per minute. And you do the same for this one here. And what you see when you calculate that is that the normal heart rate is 78.9. Now it says give the answer to the nearest whole number, so you need to round that to 79. The rate for the tachycardia is 125 beats per minute, and then you do 125 minus 79, which is 46, divided by 79, which gives you a percentage increase of 58%. So using the information, so comparing the different figures on the previous page, what are the other key features of tachycardia? You can have either that the peak with the R wave is lower, or that the P and the Q are equal in height, or that the width of the T wave was greater. Unfortunately, this trace has not come out particularly well. You can just about see it here, but it's not come out particularly well. Hopefully you have it in a better form in your paper if you're looking at it, and if you look on the website, then there's a better one on my website where you can download the completed paper. What conclusions can you draw about the way in which the person's heart is functioning abnormally? You've got to connect the P curve to the lack of atrial depolarization. You've got to link that to there be a weak atrial contraction, therefore less blood would be forced into the ventricles, and the ventricles contract normally, but less blood would be forced from the heart because there's a lack of atrial depolarization, because you can't see the P wave working as well in that ECG. So the gross anatomy of the liver, we've got a vessel coming in, and this vessel is coming, and this is the hepatic portal vein. And the reason why it's the hepatic portal vein is because it's got a wide lumen. B is the hepatic artery, narrow lumen, and A is the hepatic vein. So it says identify with reasons which each of the blood vessels. So it's not sufficient just to name the blood vessel, you've got to say why you are justified in naming that as that particular blood vessel. So it's the hepatic vein as the blood is exiting the liver, which is A, the hepatic artery as the blood is entering through a narrow vessel, so you're referring to the diameter of the lumen of the artery, and the hepatic portal vein as it's entering with a large lumen and through a branched vessel. So this leads on to the production of urea by the ornithine cycle, and this occurs in the liver and uses the mitochondria. So formation of urea, so we've got ornithine here passing into the matrix of the mitochondria where it's being joined with ammonia and carbon dioxide. Now the reason why it moves in is because it's being turned into something else, which is citrulline, and that maintains the concentration gradient so that the ornithine is diffusing in because it's being turned into citrulline. Now the citrulline is being made in the matrix of the mitochondria, so it's therefore at a higher concentration in the matrix, therefore it diffuses out. So this question is combining your understanding of the liver with also your understanding of facilitated diffusion and why facilitated diffusion would have to occur, and what would power it, which is the transformation of ornithine into citrulline, which is occurring in the matrix. So identify organelle D, it's a mitochondria, and then onto, during the cycle, ornithine moves into organelle D and citrulline moves out of the organelle. Suggest the method by which these molecules move into and out of the organelle, and then give reasons, justify your choice. So don't just state a method, state why with justification. Suggest means apply your previous understanding of theories. So it means you haven't been taught the method by which ornithine and citrulline move in and out of the organelle. However, you have been taught the concept of facilitated diffusion. So you should be aware that the transformation of ornithine into citrulline will maintain a concentration gradient. and that concentration gradient means that facilitated diffusion is possible. So here we have the method is facilitated diffusion, and why? It's that when you convert ornithine into citrulline, it gives you the concentration gradient. The molecules will not be lipid soluble, so they would require a protein channel to cross the membrane. Alternately, you could say that it was active transport, that it would need to be moved in and out of D faster than you could achieve it through diffusion. Next, how has the ammonia that's been used in step 1 been formed? And this is something you just have to know, and that's due to the deamination of the amine group from the amino acid. Then on to identify compound X. Well, compound X is made into AMP, adenosine monophosphate, so compound X is a TP, that is adenosine triphosphate. On to a part of a question which tests your understanding of how to carry out good practicals. So it says it has a high metabolic rate. It says that hydrogen peroxide is the metabolic product produced. It needs to be removed in order to prevent damage. So hydrogen peroxide is detoxified by the enzyme catalase. So catalase is the enzyme, and catalase is helping with the catalysis, that is the breakdown of hydrogen peroxide into water and oxygen. It then tells you the catalase has a very high turnover number, which is the number of reactions that the single molecule can catalyze, which is approximately 6 million hydrogen peroxide molecules every minute. Where do you find the catalase? You find it in peroxisomes, and peroxisomes are organelles which have a membrane around the outside which keep them separate from the cytoplasm. So the activity of the catalase was investigated in the laboratory using chopped up liver tissue. Now, whenever you get something of a biological origin, it's worth remembering that the concentration of the catalase will vary in that tissue and dilute hydrogen peroxide, which is the substrate. Effective increasing enzyme concentration. So they did an experiment here where the independent variable was the concentration of the catalase, and the dependent variable they've then converted into a rate of reaction because they've looked at the evolution of oxygen per unit time, which has given them a rate of reaction. Now, two variables that you need to control in this investigation. So pH, and I would add pH of the hydrogen peroxide and catalase solution, the temperature of the hydrogen peroxide and catalase solution, and then the substrate, which is the hydrogen peroxide concentration you would need to control. Now remember, you're varying the enzyme concentration, so you can't control that because that is the independent variable. Then leads on to how could you control one of the variables that you've identified. So if you've identified pH, then you might say that you would take a pH reading to ensure that hydrogen peroxide is the same for all the concentrations. You might additionally wish to say that you could add a pH buffer to maintain a constant pH. If it was temperature that you'd refer to, then you would say that you would do all of the experiments at the same temperature, and you could do that by using a temperature controlled water bath. And if it was substrate concentration, you would ensure that you'd use the same concentration and volume of hydrogen peroxide for everything that you test. So this says using the information given in part C. Now the crucial clue that they give you there is that the catalase is contained within peroxisomes, which have a single membrane around the outside of them, and they're in the cytoplasm. Deduce why and how catalase is regulated. So regulated is how much of that catalase is present. And points that they're looking for here. Why you need to regulate it is because the liver cell is going to produce large quantities of hydrogen peroxide, and the problem is if you had a very high concentration of catalase, would mean you would produce lots of oxygen very quickly, and it would be a very vigorous reaction. Therefore you have to control the rate of the catalase, and the only way you can control that, because you can't control temperature and you can't control substrate concentration, so the only way you can control the rate of the catalase catalysis is by controlling the enzyme concentration. How you can achieve that is that you can isolate the catalase in peroxisomes. Therefore, by releasing it from the peroxisomes, you can control the quantity of catalase that's present, and you can do that by releasing it in small quantities. Alternately, you could have mentioned that cells can limit the expression of the catalase gene. Therefore, you're releasing more of the enzyme if you're carrying out more transcription and translation. Now, both of those things limit enzyme concentration and therefore reduce the reaction rate. Therefore, the reaction would not be occurring too rapidly and lots of oxygen would not be produced in the liver cells. So this question is about compensation point, the balance between photosynthesis and respiration and where that occurs in plants and what times of the day. So it tells you that plants photosynthesize and respire, and it shows you a figure here with carbohydrate production and the rate of the use of carbohydrate by respiration. Now, the rate of photosynthesis starts off at 0 and is 0 during the night until the lights come on during the day, at which point the rate of photosynthesis goes up. And this is between 6:30 and about 4:30 in the afternoon. And we would assume that by about 4:30 it's then dark, so the rate of photosynthesis has fallen to 0 because there's no input of light into the light dependent reactions, so you can't power the Calvin cycle. The rate of respiration fluctuates less. It rises during the day and then it falls off later in the night. And this may well be due to the effect of temperature, in that you would expect there to be some change in temperature during the day. Even in England it does get a bit warmer during the day than at night. Then they've pointed out 2 points, L here and here, which is where the rate of production of carbohydrate is equal to the rate of consumption of carbohydrate by the mitochondria in respiration. So explain the shape of the curve for the rate of photosynthesis. So the points that you're going to have to look at here are: why is there nothing during these periods? That's because it's dark. Why does it increase between here and here? And that's because light intensity increases. Why does it peak here? Because you've got a peak of light intensity and temperature. And then why does it fall off at this point? It's because the rate of photosynthesis will be reducing as the light intensity reduces. So the marks they give you are here. There's an increased photosynthetic activity when there's more daylight, and as there's a greater light intensity, there's an increased activity of the light dependent reaction. Crucial to, when you explain, go into detail about this, so that's why they wanted that in that answer. Explain the shape of the curve for the rate of plant respiration. Well, during the day it's generally warmer, so the temperatures increase, and as the rate of respiration increases with the increased temperature. And then the explain part is that respiration is an enzyme controlled reaction, that the Krebs cycle is controlled by enzymes, and that those enzymes are affected by temperature. So as you increase the ambient temperature, the rate of respiration will increase during the day. Then it says what is happening at the points indicated by the letter L, and I mentioned this before, but at L you can clearly see that at L the rate of respiration is equaling the rate of photosynthesis. Therefore the input of carbohydrate into respiration is equal to the production of carbohydrate from photosynthesis, and that's known as the compensation point. But it's not sufficient just to say that it's the compensation point. What you have to say in explaining it is that the carbohydrate produced by photosynthesis is equal to the carbohydrate being used in respiration. This leads on to a question about the effect of temperature on photosynthesis. So it says that plants grow successfully in temperatures that are suited to their metabolism. Now this is a big clue for later in the question. Some plants are adapted for growth in cool climates, whether others grow well in warm climates. Plants vary in their photosynthetic metabolism. Some produce a 3-carbon compound as the first product of carbon dioxide fixation, so referred to as C3 plants. Others produce a 4-carbon compound, so referred to as C4s. C3s, barley, lentils, rice, soya, sunflower and wheat, and C4, things like maize, millet, sorghum and sugarcane. Then they give you a graph which shows the assimilation of carbon dioxide by the different crops. So you've got the C4 crops, these ones which are up here. So what you can clearly see is that as you increase the temperature, which is the independent variable, the rate of the assimilation, so as temperature increases, the assimilation of carbon dioxide, which is kilograms per hectare per hour, so it's a rate because it's per unit time, that increases. And the general trend you can see is that the C3 plants, which are these two here, so that's C3 and this one is C3 as well, that those plants assimilate less carbon dioxide than the C4 plants, which are here at higher temperatures. But the general trend that you can see is that as you increase the temperature, the assimilation of carbon dioxide also increases. And that's because of the action of rubisco, which is the enzyme in the Calvin cycle. So with reference to that figure, what's the general relationship between increasing temperature and the assimilation of carbon dioxide? The general relationship is that for all the crops, as you increase the temperature, you increase the assimilation of carbon dioxide. However, at higher temperatures, the assimilation decreases, and you can see it falling off for all of the plants here, here, and here as the temperature increases. It now asks you to calculate an average for the C3 plants and an average for the C4 plants at 20 degrees. So for the C3s you add them together, divide by 2, and the C4s you add them together and divide by 2, and you get answers of 34.5 and 73.5. And that is in kilograms of carbon dioxide per hectare per year, and it asks for you to include the units, so if you don't include the units you lose a mark. Then it says suggest a conclusion that could be drawn from the mean values is that C3 plants assimilate less carbon dioxide than C4. Then it asks you to refer to that table and then suggest which curve could correspond to the following crops. So sugarcane, which you grow in warm climates, you'd expect to see it as a C4 plant. And then barley, which grows in cool climates, you'd expect it to see as a C3 plant. So this question refers to temperature and their effect on photosynthesis, and temperature stress concerning plant physiologists because of climate change leading to higher temperatures. High temperature stress is the rise in temperature that is sufficient to cause irreversible damage to plant growth. And then moderate temperature stress causes these effects. So deactivation of rubisco, which is the enzyme in the Calvin cycle, that's caused by an increase in temperature and changing in the bonds that hold together the tertiary structure, the standard sort of enzyme answer for the effect of temperature. However, there's no change in the chlorophyll in photosystem 2. Remember, photosystem 2 is the one that carries out photolysis to produce the electrons, which then flow along the electron transport chain and join with NADP to form NADPH. and a reduction in stomatal aperture, which would be there to prevent the excess loss of water. Severe high temperature stress, photodeterioration of the chlorophyll breaks down, and then changes in the ultrastructure, which is the internal structure of the chloroplast, which, remember, is dominated by the thylakoid membranes and the lamellae. Assess the impact of moderate stress on the effect of on the process of photosynthesis. So this question is getting you to explain how these factors could affect the rate of photosynthesis. So you need to think about the change in rubisco and how it could affect photosynthesis, and the change in stomatal aperture. So the things that you got marks for here were to link the deactivation of the rubisco to the reduction in the light independent reaction, because you're going to reduce the fixation of carbon dioxide as it joins with the 5-carbon IUBP to form the either 3-carbon or 4-carbon compound, because the rubisco is what catalyzes that step. The reduction in the stomatal aperture, when you reduce that, then you're going to reduce the carbon dioxide that's available to the spongy mesophyll to then diffuse into the palisade mesophyll to be used for fixation in the Calvin cycle. So the key to this was to link the stress effects, the high heat stress effects, to the theory of the Calvin cycle and photosynthesis. Okay, suggest 2 ways in which the ultrastructure of the chloroplast can be altered by high temperatures. So this is getting you to think about membranes, kinetic energy, the effect of temperature on a chloroplast. And then for each suggestion, explain the effect. So tell us about the theory and tell us about how that temperature effect will alter photosynthesis. So you've always got to link your suggestion to what will happen and why. So first of all, you could have had damage to the chlorophyll or reduction in the quantity of the pigment, which affects the light dependent stage. And you could then go on to say that the membranes could be damaged, which will reduce the light dependent stage, that the damage to the membranes might affect the electron transport chain, that will reduce photophosphorylation, or cyclic or non-cyclic photophosphorylation, which will reduce the ATP production, and that the damage to the membranes around the chloroplast might lead, around the outside of the chloroplast, might lead to a release of the enzymes, which will reduce the light independent stage. You could pretty much make any reasonable suggestion linked to the effect of temperature, the damage to the chloroplast, and then linking that to a reduction in the light dependent stage or the Calvin cycle. So question 19 says, following their formation, assimilates are transported throughout the plant by translocation in the phloem. Now, assimilates is whatever is the products of photosynthesis, so this could be carbohydrates in the form of sucrose or amino acids. So phloem sap consists mainly of carbohydrate in the form of sucrose, but also other solutes. So why is it beneficial? So this is a suggest question, so this means that you haven't been taught specifically why the carbohydrate should be sucrose. However, you should be able to think carefully about what might be the benefit to the plant of transporting sucrose rather than something else. And the answers they were looking for here were that sucrose is soluble, so it can be transported in the sap, but it is metabolically relatively inactive, so it's not going to be used during the process of transport. Then on to a comparison of similarities and differences between the phloem and the xylem, and this is something you should definitely have learnt and have been taught. So the similarities are that they both carry solutes in solution, they both carry mineral salts, and they both use mass flow or generated hydrostatic pressure. Differences, however, between the xylem and the phloem, and you should be making this as a comparative statement, that the transport in the phloem can take place in both directions, that means up and down the plant, whereas transport in the xylem only takes place from the roots up to the shoots. Another comparative difference would be that the phloem carries carbohydrates and the xylem does not. The xylem carries water and mineral ions. Another difference would be that phloem transport is through living cells and transport through the xylem is through dead cells. And another difference would be that the phloem uses the capillary action and adhesion between the polar water molecules and cohesion between the water molecules and the lignin in the xylem, and the phloem does not. Now, assimilates are loaded into the phloem at the source, so things like sucrose is loaded at the source and then transported to the sink. Now, a sink is an area of net consumption of the products of both synthesis, and a source is an area of net production. It then says, explain with a suitable example how some parts of the plants can act both as a source and as a sink. And this means at different times of the year or different times of the day. How can a single part act as both a source and a sink? And the answer to this is that parts of the plant can store and then release carbohydrates. So when it's storing it, it's acting as a sink, and when it's releasing it, it's acting as a source. And a suitable example might be a root or a leaf, or a storage organ such as a tuber, which can act as a sink or a source at different times of the year. So this asks you to explain the process of the loading of sucrose by referring to what's happening at A, B and C. So what we have here is B is the active loading of sucrose into the sieve tube element, which is going to lower the water potential in the sieve tube element. The way this is achieved is through the active transport of protons, and the protons are moved into the companion cell by the use of ATP. So ATP is being hydrolyzed, and that's moving the protons from where there are fewer protons to where there are lots of protons. So there are lots of protons in this companion cell. Now those protons then move back into down their concentration gradient, moving back into the sieve tube element, and as they do that, they move a sucrose against its concentration gradient. So the gradient of the protons, which have been actively transported from here into here, requiring energy, those protons then flow out again through a co-transport pump, moving the sucroses to an area where it's more concentrated inside the sieve tube element. Now that movement of the sucroses creates a water potential gradient, so water moves by osmosis from a xylem cell, which is C, and also from the companion cell, which is A, into the process shown at A, and both of those create a hydrostatic pressure, and that hydrostatic pressure then forces the phloem through the holes in the sieve tube, in the sieve plates, into the next cell. So it physically moves the phloem through the sieve tube element. So what they were looking for here was used to specifically say what is happening at B, at A and at C. And what's happening at B was that the sucrose is being pumped from the companion cell into the sieve tube by active transport, use of the co-transport of sucrose, reducing the water potential of the xylem, leading to water entering at A, water entering at C, which increases the hydrostatic pressure, which is going to force the sap down the sieve tube through the pores in the sieve plate. So on to a data analysis experimental question. So it shows you a diagram of a potato, and potatoes are tubers, which are underground storage organs. So the point that's going on here is that the products of photosynthesis in the source which is the mature leaves, which are taking in carbon dioxide and joining them together with water to produce carbohydrates. Those substances are being transported down into the storage organs, which are the tubers. So it says actively growing tissues have a high demand for carbohydrates. This means that a lot of the phloem sap is directed to these tissues, and these require sucrose to be unloaded in large amounts. So in an investigation, potato plants were modified by having a gene for invertase inserted into their DNA, so that the gene for invertase would be expressed. And expressed means turned into the protein, which is the enzyme invertase. So invertase is responsible for catalyzing the hydrolysis of the disaccharide sucrose. Remember, sucrose is a disaccharide of glucose and fructose joined together by a glycosidic bond. So a trial experiment was carried out to compare the properties of the modified plants with those that have not been modified. After harvesting, the tubers of 3 of the types of plants were compared. So 3 of each type of plant. You might think maybe that's not enough to compare, and worth thinking about that they've told you it's only 3. And here are the results. So when you get a table like this, firstly have a very good look at it and make sure you think about the data before you get distracted by the question. So mean number of tubers per plant, well, there's a big difference in that there are fewer tubers in the modified than in the not modified. They're about half the number. The mean mass of each tuber, however, is much greater. So it's about 3 times the mass in the modified than in the not modified. Big difference in the sucrose concentration. It's about 9 times less in the modified than in the unmodified. The mean glucose concentration is massively higher than in the not modified, and the variation in the mean glucose concentration is also much higher. And the invertase activity is 60 times higher in the modified than in the not modified. So all of this data is going to be referred to in the next part of the question, in the last part of the question. So firstly, name the bond that is hydrolyzed, and it is a glycosidic bond, which is the bond between the glucose and the fructose. So that is important that you name it as glycosidic. So this now asks you that the potato tubers contain monosaccharides. Compare the concentration of the monosaccharides, which is the glucose, in the modified tubers with those which were not modified. Now compare means you must do a calculation and you must express that calculation, and you would say that it was 19 times greater in the modified than in the unmodified, or you could have worked out a percentage increase between the modified and the unmodified, and also you should have referred to the standard deviation in the concentration of glucose. Then it leads on to in the modified plants, the unloading of the sucrose is increased in the tubers compared to those which are not modified. Transport of sucrose to the tubers was also increased in the modified plants. Using the data and the information given, so you must refer to the data that's given in the experiment, deduce a possible mechanism to account for the increased unloading and transport of sucrose in the modified plants. Now, what they're looking for here is for you to realise that the sucrose is being unloaded at the sink, which they've told you that the tuber is a sink, and that the presence of the invertase, which is the enzyme caused by the insertion of the invertase gene, is going to convert the sucrose into glucose, or a monosaccharide, which is glucose and fructose. And that's going to increase the sucrose concentration between the phloem and the sink. It's going to increase the sucrose concentration gradient, between the phloem and the sink, because in the sink, the sucrose is going to be hydrolyzed by the invertase into 2 monosaccharides. That's going to cause an increase in the concentration of sucrose increase in the unloading of the sucrose from the phloem. How that could help is that that's going to increase the solute gradient between the source and the sink, because in the sink the solute, which is the sucrose, is being converted into something else. the water that's going to be leaving from the phloem is going to increase the pressure gradient, and all of that is going to increase the movement between through the phloem, through the sieve tube element. Okay, then it goes on. The trial experiment compared the properties of the modified potato plants with those that were not modified, analyse the data and draw conclusions about the yield of the tubers. Now remember that the modified plants produced fewer tubers, but those tubers were much larger compared with those tubers from plants which had not been modified. So the yield is the number of tubers multiplied by the size of each tuber. So what they were looking for here was that you had realised that, and realised that the modified produces fewer and larger tubers, and that the modified produce a total mass of tuber which is greater than the other modified. And if you'd multiplied the tubers by the number of each tuber, the size of a tuber by the numbers of those tubers, you come out with those data, which is about 109 and about 89. So you see that there's an increase in yield for the modified compared to the not modified. On to a question about fight-or-flight responses. So here it shows a flow diagram, shows a sequence of events once a threat has been perceived. So we have the nerve impulses transmitted by the sensory neurons to the hypothalamus, and then the hypothalamus coordinating the fight-or-flight response. So you have nerve impulse transmitted down the spinal cord. They reach the adrenal glands and activate the secretion of adrenaline. The adrenaline then acts on the target cells. Then you also have, at the same time, stimulation of neurosecretory cells, which releases ACTH by the pituitary gland near the hypothalamus in the brain. The ACTH attaches to receptors in the adrenal cortex. That results in the release of cortisol, and cortisol causes an increase in blood pressure, change in blood sugar levels, suppression of the immune system, all of the parts of the fight or flight response. Then it leads on to identify two signaling molecules. So you could have picked ACTH, cortisol, or adrenaline for just 1 mark. Then on to what adrenaline then causes to happen. So adrenaline acts on a variety of cell types with a variety of responses. And then complete the table by stating the effects of stimulating each target cell. They want the target cell followed by the response, followed by why that might be helpful in the fight or flight response. So it then starts with the sinoatrial node. Remember, the sinoatrial node is in the right atrium, and it's where you start the wave of contraction that spreads out across the atria and then goes down through the atrioventricular node and stimulates the ventricles to contract. So the response would be that you have an increase in the rate of firing impulses, and why that might be helpful is that's going to increase the heart rate so the blood will circulate more quickly and carry away carbon dioxide and supply more oxygen and more glucose to the tissues that need it. What might be happening in the liver is that the liver would increase the rate of glycogenolysis. Now, lysis means to split, and then this is the splitting of glycogen. And glycogen, remember, is the storage polysaccharide of glucose. It's a branch chain storage polysaccharide, and when we increase the rate of glycogenolysis, we're increasing the glucose concentration in the blood. Therefore, we're making more glucose available for respiration to power the running away from something or fighting with it, one or the other. And then the erectile muscles in the skin. As a result of the release of adrenaline, you're going to cause contraction of the erectile muscle, and the effect of that is going to raise the hairs and make you look larger and more aggressive. This is obviously a bigger effect in animals other than us, because it's not going to have a great deal of effect in us, but it is in plenty of other animals. Okay, describe the sequence of events sequence of actions that occur once adenyl cyclase is activated in the target liver cells. So what you have to just know this is that it catalyzes synthesis of cyclic AMP from ATP. This is a secondary messenger, and that cyclic AMP then activates the enzymes which cause the hydrolysis of glycogen into glucose. Then goes on, the response also occurs when a person is subject to stress. However, the body does not need to respond physically to the stimulus, and for example, the bronchioles do not dilate. From the information given and your knowledge suggests the long-term adverse effects of continued exposure to stress on the body. So, problems that can occur as a result of the misdirection of the fight or flight response because you're suffering from stress but you're not actually having to fight or fly, is that it can lead to prolonged high blood pressure, can lead to cardiovascular problems, prolonged high blood sugar levels can lead to problems with diabetes, and suppression of the immune system leads to susceptibility to disease or infection. So part of the body's response to fight or flight is to run away from the threat. Prolonged vigorous exercise puts high demands on the body's metabolism. Muscle cells require an adequate supply of oxygen for respiration. If insufficient oxygen is available, the cells must respire anaerobically. So this is linking two different parts of this spec. So this is linking the respiration of glucose to the fight or flight responses in homeostasis and nervous, hormonal and nervous communication. So it starts off with outlines the process of anaerobic respiration in muscle cells. Remember, anaerobic respiration is just glycolysis with an alternative way of getting rid of the NADHs, which you can't regenerate by sending them off to the electron transport chain. So the step here is that glucose is being hydrolyzed. The glucose is a 6-carbon compound. It's being split in the glycolytic step to produce 2 3-carbon pyruvates, and that 2 3-carbon pyruvates cause the net production of 2 ATPs, which are used for muscle contraction in the muscles. Now that's then going to take an NAD, and that's going to be turned into an NADH. Now ordinarily that NADH would go off to the electron transport chain, donate the electrons and the hydrogens, which would go down the chain, join with oxygen to produce water. Unfortunately, you don't have enough oxygen, so you're respiring anaerobically. In anaerobic respiration, you take the NADH, and that NADH is then regenerated back into NAD, so that this process can continue with the net production of 2 ATPs. Also takes the pyruvate D, and the pyruvate is being reduced to produce lactate or lactic acid. So it says identify the compounds labelled D and E. So D is pyruvate and E is lactate. What is the role of compound D in anaerobic respiration? Well, the role of compound D is that it is accepting the hydrogens and electrons from the NADH, and that it is then, in doing so, becoming reduced to produce the lactate. And because it's enabling the regeneration of more NADs, it's meaning that the lysis of glucose can continue to produce a net production of 2 ATPs, which can be used for muscular contraction. So the answer is it's a hydrogen acceptor. Then why is it important that compound G is formed during the reaction in which D is converted into E? And the answer to this is that in order for you to have glycolysis, you need NAD. In order to regenerate the NAD, you need to reduce the pyruvate to lactic acid because there's a limited amount of NAD in the cell. Because you're regenerating the NAD from the NADH means glycolysis can continue, and that glycolysis produces a small quantity of ATP, which enables muscle contraction to continue to occur. So compound E is toxic and is removed from the muscle cell. It's transported to an organ in the body. Which organ is compound E transported to, and how does it reach the organ? Well, it's transported to the liver, and it's transported in the blood. So athletic sprinters can require large amounts of energy in short periods of time. They require large amounts of energy to power muscle contraction. Under normal conditions, exercise requires an increased rate of breathing. It's been observed that some of the best sprinters take only one breath at the start and do not inhale again until the end of the race. Suggest how these sprinters can expend so much energy without needing to carry out aerobic respiration. So suggest means you might not have been taught about this, but you should apply the principles which you understand about anaerobic respiration. And the answers that they're looking for here is that the cells, that their muscle cells, can tolerate high levels of lactate or a low pH or high levels of acidity, which is produced by the lactic acid. Do they have high levels of phosphocreatine? They can use stored ATP. So I hope all of that's been useful. Well done for making it to the end, and best of luck with your exams.